Okay, in this part of the video, we're going to kind of look closer at the DNA and take a look at some of the enzymes and how replication begins. So you have this picture of the DNA molecule, the double-stranded DNA in your notes. Let's start by identifying some of the key parts that we just did before. I'm going to start by labeling the carbons on this particular sugar molecule. Remember that the base attaches to the first, and then you go around clockwise. Okay, and then the fifth carbon is actually up here. This is an oxygen. Okay, so that means that this end would be considered the 3' end. And since here's your 5' here, this is the 5' end. So now that I know that, this is the 3' end, and this is the 5' end. This orientation is super important because this is determining the direction that replication or elongation will happen. The other things that we want to understand is the base pairing rules. T bonds with A, G bonds with C, and so on and so forth. We also want to understand that these bonds holding the bases together are hydrogen bonds, and that those are weak bonds. You can break those apart relatively easily, which is also important in DNA replication. And then the last thing I want to understand, or make sure you understand, we already talked about how these were covalent bonds, but we can further specify that these are the bonds within a nucleic acid, within DNA. So a long time ago, we identified that these are phosphodiester bonds, which are still a type of covalent bond, but this indicates that you're talking specifically phospho, all right, the bonds in the backbone of DNA. All right, now it says the remainder of the drawings are going to be much more simplified structure of DNA, and we're really going to focus on eukaryotic. So let me pause for a second and look at this picture here, and let's think about how eukaryotic and prokaryotic DNA are similar, but also how they're different. They're both double helix, okay? They both would contain ATCG. They both would have those sugar phosphate backbones, but the big difference is that eukaryotic DNA is linear and prokaryotic DNA is circular. So when it comes to replicating DNA, notice how in a prokaryotic DNA, there's a replication fork, meaning we're copying the DNA here and here. And then when complete, you'd have 2 circles of DNA. Whereas in eukaryotic DNA, we have what we are going to call replication bubbles. Each would have a fork, okay? But then this is a bubble with 2 forks. This is a bubble with 2 forks. So replication happens in multiple places along the DNA for eukaryotes. Again, for prokaryotes, this would be like the bubble with 2 replication forks. Okay. So again, we're looking at eukaryotic DNA. There's a couple of things we want to point out. Helicase. We're going to use these little triangles to represent this enzyme. It's breaking those hydrogen bonds that are holding those bases together. Okay, so each of these little colored pieces sticking out is a base, and then that means that this is your sugar phosphate backbone. Okay. So again, a much more simplified model, but easier to keep track of. That also means that if this is the 5' end, if I trace this, that means that this is 5 and this is 3. Here's the 5. If I trace back this strand, okay, here's 5 and here's 3. Okay. This becomes important because we think about the direction in which replication is happening, which we will get into the details in just a few minutes, but we're just getting a lay of the land here. There will be proteins, and we'll see this in a minute, that bond to the DNA these single strand binding proteins that help keep the DNA apart while it's being replicated in between. All right. Now, we're going to look at elongating the DNA, and we need a particular enzyme to be able to do that. So we've talked about helicase right here. Now we're looking at DNA polymerase. So DNA polymerase is an enzyme that will basically be adding free nucleotides to an existing strand. This is endergonic, so it actually does require energy to be able to build this DNA molecule. Nucleoside is basically a sugar plus a phosphate, sugar plus a base. So we're identifying that it actually has 3 phosphates attached to it. If you remember from the DNA structure and history notes, we said that these nucleoside triphosphates come in, and as a result of that, when they join in, 2 phosphates break off, and that process releases energy. So that's freeing up energy that allows DNA polymerase to add these new nucleotides on. All right, when I look at this picture here, again, let's kind of figure out what we're looking at. These little tick marks represent a base. The solid line running through is the backbone. These little circles are these single-stranded binding proteins. Like I mentioned before, they help keep DNA separated while we are adding that complementary strand. Right. Here's helicase. Remember that he's responsible for breaking the bonds, the hydrogen bonds, holding the bases together. And this is a new one that we haven't talked about called topoisomerase. I have a video clip on the links page that looks at how he works and what he's doing, but basically he helps with the DNA becoming overwound, because DNA is a double helix. When you start unzipping it, there's a lot of tension that builds up ahead of the replication fork. So topoisomerase will help relieve some of that overwinding. So there's a video clip on the links page if you want to go look at that. Okay. So a couple more things that we want to look at. This enzyme, Primase, is also known as RNA polymerase. So previously, we just mentioned that DNA polymerase adds new nucleotides, but it adds DNA nucleotides. So RNA polymerase is needed because what has to happen first is that we have to place an RNA primer. Okay, that's what this is right here. We're placing some nucleotides of RNA onto the parental strand. Okay, so these template strands that we've separated. So notice that this is an RNA primer here, and this is an RNA primer here. And their locations are different, and we'll get into that in just a minute. But our other focus is on DNA polymerase. So we kind of drew it in as a dashed box here so we didn't cover up everything, but this is your other enzyme, DNA polymerase. Okay. DNA polymerase is going to attach DNA nucleotides to the 3' end of the RNA primer. Now, you might be thinking, why do we have to put an RNA primer down? Well, this enzyme, DNA polymerase, needs something to attach nucleotides to, whereas RNA polymerase, also known as primase, does not. It can place nucleotides down without anything being already there. So we have to place this, what we call this primer, down so that RNA polymerase, sorry, so the DNA polymerase can add in these nucleotides. Also remember, elongation happens by adding nucleotides to the 3' end. So that means that you're going to add nucleotides in this direction on this strand, but add nucleotides in this direction on this strand. So DNA replication happens in opposite directions depending on which strand you're looking at. Okay, I'm going to stop the video right now before I get into the details of the next part.